up we have Team Grenoble Alps.
Alright, people! My name is Thomas. I'm here today with Elise, Fanny and Charles. And first, let me tell you that we are so happy to be here today and to have this opportunity to present to you our project, Phagyzer.
Okay, so first, to contextualize a little bit the project, we wanted to fight an issue that our society is facing today, which is antibiotic resistance. As you might know, today, around 700,000 people die each year due to antibiotic resistance. And that's not it, because in 2050, according to the WHO, the World Health Organization, there will be around 10 million people that die each year. Moreover, the WHO published a list of the most pathogenic bacteria and regarding antibiotic resistance. And the top 3 we could find Pseudomonas aeruginosa, and this is why we wanted to focus on this bacteria. With all of this knowledge, awareness is raising, and with it, people are thinking of new alternatives to face this problem. One of them is phage therapy, and this is the one we wanted to focus on in our project. Phage therapy is the use of bacteriophages to treat people that have infectious diseases. What is phage therapy? So a bacteria integrates its DNA into bacteriophages integrates its DNA into the bacteria, and this triggers multiplication in it and bacteriolysis, and this kills the bacteria.
Alright, so bacteriophages are really good at killing bacteria, but the problem is that they are really, really specific, and so the doctor doesn't really know which phages to give to his patient. And that's where our project comes in. Our idea is a fully automated device, able to give to the doctor 2 main information. First, the identity of the bacteria, and then to select the most effective bacteriophages against it. We also wanted to give to the doctor a third information, which was whether or not there was an antibiotic resistance marker inside the bacterium DNA. Once we had this idea, we wanted to confront it to our society. And actually we are quite lucky, because even though Grenoble is a quite small city, we have the big scientific community. Our first meeting was with patient. For 10 years, he used antibiotics to try to cure his feet infections. And after 10 years, none of the antibiotics he was given did work. So what he did was trying to use phages to cure his feet. And after 6 months, he was really doing well. He was doing better, and he kept his feet thanks to phages. We also went to doctors, Dr. Morin and Dr. Epola, who are the head of the infectious disease department, and they told us that they are facing antibiotic-resistant bacteria every single day. For them, phage therapy was really an interesting alternative, and they liked our idea very much. At this point, we figured out that biologists and engineers couldn't do this project alone. We needed to take a step back. And that's when we met Loïc Lasek. Loïc is a wonderful guy. He's a paramedic, but also has a bioethic formation. And he helped us take a step back on the project and on our human practice. We like him so much that we integrated him in our team, and he's here today with us as an advisor. We then went to the biggest medical analysis lab in our region, Novel. And we were actually quite surprised, because all of our tests are done on petri dishes. And for them, our system was really relevant, and they told us to keep in mind the regulation and laws about biomedical devices.
We also had the opportunity to work closely with Biomérieux. It is a top 10 French company, and they work in the in vitro diagnostic field. They really liked our project, and so we decided to work with them all around the year for our project. They gave us so many advices, and for example, they gave us some materials and they also give us some space in their laboratory in order to practice the experiments on the bacteriophages. We also went to universities in order to talk with the future pharmacologists and practitioners to talk about this kind of therapy, which is phage therapy. And we also carried out a survey in order to see if the people are aware about phage therapy, and more importantly, they are open into it. So this is one of the results of the survey, and we can see that most of the people will maybe use phage therapy, but there is still a lack of information around this subject. This is a global view of our proof of concept of the device, and the first step is to take a sample from a patient that might be infected by a pathogenic bacteria that we want to detect, which is Pseudomonas aeruginosa, like Fanny said. Then we add a specific phage against this bacteria, and so the bacteria died and the bacterial DNA will be in the solution and ready to be extracted. After the extraction, we want to detect a small fraction of the whole genome of this bacteria. And we do this thanks to DNA probes. The whole thing will be transformed into competent cells, like E. coli, for example. And then we measure fluorescence, and these results give some information for the practitioner in order to make the right decision for the patient health.
All right. But remember, we want to automate the whole thing. And so we need to be able to perform 4 main actions. We need to be able to pipette liquid, we need to be able to control the temperature, we need to be able to extract DNA, and finally to measure fluorescence. All of those modules are linked with a rotative plate who places the right tube beneath the module. This rotative plate right here. And you also have to keep in mind that we want to automate the thing, so we have to conserve and store biological products in our device. For that we have a lot of work on drying and freeze-drying techniques in order to achieve that.
Stage 1, Phagyzer.
Okay, so in a diagnostic context, finding a specific phage against one pathogenic bacteria is maybe the perfect solution to select the bacteria that we want to detect into a complex sample. So the very first step was to select the right phages against the bacteria that we want to detect, and we decided to work on Pseudomonas aeruginosa. So if the bacteria is actually in the sample, the phage will kill it, and then the bacteria will be released in the solution. And that's what we expected, because we want to detect the bacterial DNA. And each time the phage kills the bacteria, it amplifies itself, and this exponentially.
Alright, and to automate this first step, we need to take phages and put them in contact with the patient sample. To do that, we use an electronic pipette, as you can see here. An electronic pipette is something that is used by humans in a lab, and it's very accurate. As you can see here, we kind of hacked pipette by soldering wires directly to the switches and connecting those wires to a microcontroller, in our case an Arduino. By putting the pipette on a LiDAR stage that can be commonly found in a 3D printer, we can move the pipette up and down, and with the Arduino press the switch and pipette liquid. That's how we handle liquid in our system.
Stage 2, DNA extraction.
So the point of this step is to extract the DNA that will release after the phage action. To do so, we use magnetic beads in order to attract the DNA, and we use a magnetic field. It's just like this game. For example, we have the Pokeball. The Pokeball is the magnetic beads. It's actually catching the DNA, and then it's attracted by the magnet.
Alright, and here in terms of automating this, it's quite simple. All we have to do is push magnets close or away from the tubes, and with the pipette that I already presented to you, we can do the washing steps and extract DNA.
Stage 3, digestion.
So, after the lysis of the bacteria, the whole DNA will be in solution. But this is the whole genome, and we just want to detect a small fraction. just like this. So, we selected enzyme that we cut a small fraction, and this is the target, the specific target that we want to detect. And the target will be free and ready to be detected, and we use HAE3 enzyme that cut a fragment of 36 nucleotides.
Okay, so before taking you through all the detection process, we're going to present to you how we designed the props. So the props were inspired by a previous team, so it's the iGEM Cork team from 2015. We designed a prop which is actually a plasmid with a single-stranded fraction able to detect our target. So as our prop is so special, we had to design everything bioinformatically. So we had to find the complementary sequence of the targets. We then had to add restriction sites and overlappings. The overlappings enables the prop to be integrated. The prop to be integrated into the plasmid. This enables so the detection of the targets. To be more specific about how we designed the prop, so now that it is integrated into the plasmid, we first had to linearize the plasmid using either PCR linearization or digestion. Then we had to use nicking enzyme. So as you might know, nicking enzyme can take out only one strand of both strands of the DNA, leaving the complementary sequence of our targets single-stranded and able to detect our targets.
Stage 4, hybridization.
So now that we have the target and the prop, we can trigger hybridization using the right temperature. It is a little bit like the game Tetris. As you can see in pink, it's our target, and the circle is our plasmid. So as you know, to win Tetris, you have to fill the holes, and it's exactly the same for us.
So to perform this hybridization step, we need to reach a specific temperature. And actually, this temperature is of 93 Celsius degrees. So, to reach such a high temperature, in our device, we use the heating side of a Peltier unit that is placed next to the tubes. To make sure that the temperature is properly set, an infrared sensor measures this temperature from the top of the samples. And by the way, these samples are in aluminum buckets, so that the temperature is correctly homogenized. And it is indeed homogenized, as you can see on this video taken with a thermal camera.
So before taking you through the last step, we wanted to remind you, because there's a lot of information, all the process we went through. So the first step was to lyse the bacteria using bacteriophages. Then we had to extract the DNA of the bacteria using magnetic beads. The DNA was then digested to be able to be detected by our probes. And the last step is
bacterial transformation.
So in our case, in our project, there are two cases here. The first one is if the probe never encounters the target, as Mario is doing, we will not integrate the plasmid, and because there is a gene that permits selection, which is the chlorophyll gene, the bacteria colony will die. The other case is if we have a recircularization of the plasmid, then the transformation can occur, and we will have fluorescence occurring because our plasmid that we use has a RFP gene which enables production of fluorescence.
So, here again, the temperature control is essential. To insert the probes in the bacteria, we alterate their membranes by a thermoshock. And to realize this thermoshock, we use two blocks. One that is set to 42 Celsius degrees thanks to a heating resistance, and another one set to 11 Celsius degrees, as you can see on this graph, using the cold side of a Peltier unit. So, you can see that this thermoshock is not that wide, only 31 Celsius degrees, but that is enough to perform a transformation, as you can see on this picture. So, now that the bacteria are transformed, they can express fluorescence, and this fluorescence is analyzed thanks to our fluorescence module. It was designed to ensure dark conditions, so that the picture taken by the camera through a filter can only display fluorescence. So here is an example of fluorescence picture, and as you can see, it is not that bright. But as there is absolutely no noise, it means that the light detected can only be fluorescence. And detecting this fluorescence means tell the doctor if the patient is infected by the pathogenic bacteria.
With this measure of fluorescence, the job of our device stops, but not our project. Indeed, we wanted to give the doctor a tool for the next step. And we did a modeling system of the phage bacteria interaction. Here's one of the results of this model. As you can see here, there's a yellow curve, which is the phage population according to time, and a red curve, which is the bacteria population according to time. And the thing is that you see here there are quite relatively a low concentration of phages, but because they amplify, they will rapidly infect bacteria and amplify. And when there are too much phages, the bacteria population will start to decrease rapidly. So I would just want to take a moment to say that for us, we did this model and improved this model thanks to tight collaboration with iGEM Nottingham in England, and they were working on a model with a different kind of phage bacteria interaction. I would just want to say that for us this iGEM project was an amazing experience. We had to do a lot of things, to learn a lot of skills on the fly, hard skills, soft skills, you name it, we learned it. We also were able to present our project to a lot of people. Here that was taken a month ago, and we did a presentation in front of more than 300 people. We were also able to present to a more diverse crowd in the science fair, to other iGEM teams during meetups. We conducted a survey, an educational survey, and we created a small YouTube video on the theme of antibiotic resistance. Alright, let's sum up, shall we? So we managed to achieve a lot of our goals. We were able to do bacteriophages lysis and DNA extraction. We were able to conserve and store a lot of our biological products inside our device. And we were able to design and construct DNA probes, even though there's still a bit of characterization work to do. On the engineering side, we were able to do a fully working pipetting module. We were able to control the temperature effectively, and the DNA extraction works just fine. Our fluorescence module could be a bit more sensitive, but we think that we now have the right system, and that by changing the camera, that will be all good. And finally, the automation of our system allows us to link all of those modules without struggles. So we think that we have a strong proof of concept of an automated device, diagnostic device, promoting phage therapy. And the best thing is that it costs less than $1,300. So we think that in the future it will be a real tool for doctors and hospitals against superbugs. And we think that it has its place in the hospital near the doctors. And why not? Maybe there will be also right next to our device, the device from the iGEM Munich team, which hello guys, which were able to produce phages for phage therapy on site. I would like to take a moment to thank all of our sponsors, without whom we wouldn't be here today, and thank you all very much for your attention.
Right, first, questions from the judges. All right.